Hi, everybody, and welcome back to Miss Angler's biology class. I am Miss Angler. In today's video, we are going to be looking at plant organs. Now, it is at this point that if you are not so certain about plant tissues, you should click the video now at the top right-hand corner, go watch that video, and then come back here. The reason for that is you need to know all the plant tissues and their functions and structure before I can apply and show you how they make these organs. Now, if you are new here, don't forget to give this video a thumbs up and make sure you are subscribed with your notifications turned on because I post every Tuesday and Thursday. So let's get into the video now, breaking down our plant into two major systems. The first system is above ground, which we call the shoot system. This is where we're going to go through the structures that we see in the leaf and the stem, and then we're going to go into the root system, which of course is exclusively the roots as well as the outgrowths called the root hairs. Now remember, it's really important that we understand that there is a difference between plant organs above and below ground, and sometimes there are certain structures or tissues that have slightly different roles above or below ground. Now, before we go any further in this section, we do need to recap the three main tissues I'm going to talk about throughout this video, and I'm going to apply these tissue types to each of the organs so you know how they work. Now, remember, these are organs, which means they need to be made up of many different tissues working together for a common cause. So the first group of tissues that we want to go over are going to be the dermal tissues. Now, you may remember the dermal tissues as these are the tissues that are responsible for covering the outside of our plant. We see this on the stem, the leaf, and the roots. Now, these dermal tissues can be specialized, as we see as our example here. We can get something like stomata, which is a special kind of dermal tissue, and it is adapted for a very specific function, but I'll get into that later. The next kind of tissue type we need to just quickly revise over is vascular tissue. Now, vascular tissue comes in two kinds, and vascular tissue is either going to be xylem for water or phloem for food. It's also important to remember that this is what gives the plant structure and support. It's also the transportation tissue, and as we can see with the arrows in this diagram, xylem goes in one direction from the roots up only, whereas phloem can go both down and up because it's transporting food. Now, the final group of tissues we are going to look at is ground tissues. Please don't be confused by this name. It doesn't mean that these tissues are in the ground or underground. They are simply the building tissues, or they provide the foundation that we build our plant off of, hence ground, building from the ground up. And these tissues generally fall into three major categories. They are parenchyma, collenchyma, and sclerenchyma. Please remember, you do need to be able to draw and label and tell the difference between these tissues in exams and tests as well. And you also need to know the differences in their functions based off of their structures. Now, when we just quickly go over their different variances for their functions, remember that parenchyma is the packaging tissue, so it's big cells. We've got collenchyma, which is the structural support, but not as hard and rigid. Generally, we see this in a stem. It's often green and filled with chloroplasts as well, and it's got thickened corners. And then finally, we've got sclerenchyma, which is the very, very hard connective tissue that we have in plants. It's what forms things like the husk of a seed or the bark and the wood of a tree, and it has very thickened cell walls all the way around it. Now, each of these tissue types are going to be explained for each of the organs. In other words, for every plant organ, you must know the dermal tissue that is present, the vascular tissue that is present, and the ground tissue that is present. And it varies from organ to organ. So let's begin with our first organ of a plant, which is, of course, a leaf or leaves. Now, leaves are really important because they are like our solar panels, right? They are trapping the sunlight, they are absorbing it, and they are using it for photosynthesis. And they've got a lot going on inside of them, each of them having a very important function. Now, this is a diagram of a 3D image of a leaf that we have cut open so we can see it on the inside. And I'm starting off with something called the cuticle. Now, the cuticle is a see-through translucent covering, and its most important function is that it is there for waterproofing. Now, waterproofing is really important because you need to prevent water loss due to heat, and that's what the cuticle does. It's also clear, which means that it allows sunlight to move through the cuticle and into the epidermal layer, which we see here now. And that brings me to the next layer, which is, of course, the epidermis. And the epidermal tissue, if we remember, is the tissue that is responsible for protection. Now, the upper epidermal layer doesn't necessarily have any color to it either, and it often is there as a protective barrier. The most important thing about this also is that it is one cell layer thick, so it's not a very thick layer, but it acts as the skin or outer layer. Now, sitting just below that is something called the mesophyll. Now, the word meso means middle, okay? And the mesophyll is made out of two kinds of special cells. One is called a palisade cell, the other is called a spongy cell. Now, we call it a palisade cell because, like a palisade fence, all of the cells are elongated like rectangles and they sit side by side. The spongy cells, or the spongy mesophyll, are the irregular shaped cells that sit just below it. Now, their shapes are really important in the way that they are put together because it influences, of course, their function. So let's just quickly speak about palisade cells. Palisade cells are stacked the way they are. They're stacked standing up because you want to be able to put as many cells next to each other as possible. Now, what's also really cool about palisade cells is that the chloroplasts can actually move inside the cells. And so what they often do is they accumulate near the top end of the cells so they can be closest to the sunlight. Spongy mesophyll cells, which are these cells that are lower down and on our diagram, they are these lighter green cells that we see over here. They are more of the packaging tissue. They are a type of packaging tissue that is used often for water and for water evaporation. But I'm going to get more onto that in another video where I go into the transportation and support systems of plants. The next thing that we see inside our leaves is, of course, the next group of tissue, which is our vascular tissues. We have got our xylem and we have got our phloem. Now, remember, these are running through veins in leaves, which I'm sure you've seen before, and that's what this entire structure is over here. What are they transporting? They are transporting all the products of photosynthesis. So when we are photosynthesizing, we are taking all of those carbohydrates and we are moving them around the plant, and that is moving in the phloem. The xylem, on the other hand, is bringing water all the way up from our roots to the leaves so that we can use that water to photosynthesize, and that water is going into our mesophyll. Now, our final underside of our leaf, again, has a repetition of structures. We've got the lower dermis and the lower cuticle. So this is on the underside of the leaf. But now we've got a really important extra modified epidermal tissue, which is this over here. It is the stomata. Now, the stomata is a modified epithelial cell, and you actually have two cells that sit side by side, and they look sort of like jelly bean shapes. And what happens is they have really, really large vacuoles on the inside. So this little black area here is going to be my vacuole. And what we do is we fill that vacuole up with liquid, or we drain it to make it open or closed. Now, right now, this stomata is open, whereas this stomata would be closed. And that's how we open and close the stomata to allow specifically gases, but also water to leave the leaf as byproducts of photosynthesis. Now, all of this is going to be even more elaborated when I move on to transportation and support within plants. The next structure I want to move into is stems. Now, there are more than one kind of stem. Actually, there's a monocot stem and a dicot stem. I'm specifically going to focus in on dicot stems. It depends on the school and the curriculum that you're doing, but the majority of us are only going to learn about a dicotyledonous stem, which is what you see here. And I automatically already know it is a stem for a couple of reasons. One, of course, before we go any further, look at this shape, okay? This is a cross-section through a stem, and we know that stems are generally like a cylinder, right? The next thing I know, and I can confirm that this is a stem, is because of these egg-shaped bundles of tissues. They sometimes look like eggs, or maybe sometimes they also look like an acorn from a oak tree. Depends on how you see it, but that is the defining thing to say this is a stem. Now, why am I making such a big deal about this is because when we go on to roots just now, you might confuse stems with roots. So this is what it looks like. We have little bundles of tissues in a circle around the center, and that's how I know I'm dealing with a stem. Now, let's go into the various tissues. Again, every single one of the tissues we've covered must appear here. So starting off with the epidermis. Now, a stem does have an epidermis, and this epidermal tissue sometimes contains chloroplasts. Often it is in the collenchyma, which sits just below the epidermis, and that sits inside the cortex. Now, the cortex is the filling of the stem, and as I said to you now, it can contain collenchyma. Now, if collenchyma has chloroplasts in it, we call it chlorenchyma, and that's what gives a stem a green look to it. And so I want you to imagine that this whole entire area here, where it's got that stripy green detail, that is all going to be the cortex. Now, sitting beyond that are, as I mentioned to you earlier, these little units of tissue. And it is made out of our second category of tissues, which is the xylem and the phloem. And these are our vascular tissues. Now, as it may already be very obvious, stems are conductive tissues. In other words, we are moving things from the roots and the leaves up and down. And so that's why it's really important that these vascular bundles, or this xylem and phloem, appears. And so in many textbooks, we refer to these little egg-shaped structures as a vascular bundle, and that's what it is together. It's really important, everybody, that you put the phloem on the outside and the xylem on the inside. It's really, really important. So we've got the water transportation on the inside and the food transportation on the outside. It will be very clear again why that is happening once we get to transportation in plants. Now, the final two things I want to cover is this white inner ring, and it is called the cambium. Now, cambium is a type of tissue I didn't recap earlier, but you may remember this from learning your other plant tissues. A cambium is a meristem. And meristems, remember, are able to turn into things. So it means that they can change, they can differentiate. And this cambium is what is allowing the stem to grow and widen and thicken, but it can also sometimes become bark. And we'll get into that a little bit more later on, again, once we go into more detail into plants. But the basics is that cambium allows for lateral growth. In other words, taking a narrow stem and making it much wider. Now, the final tissue we're going to go over here is the pith. The pith is all this central tissue that sits in the middle here. It is mostly ground tissue. Think things like our parenchyma. It's quite fleshy and green, and as plants age and change, and maybe this stem is going to become a woody stem, the pith, the cambium, and the cortex all slightly start to change consistencies. But for now, I'm only talking about green, fleshy stems, so we'll stick to the pith being made out of parenchyma. Now we're going to move on to our last plant organ, which are roots. And roots are incredibly specialized, like leaves. And so it's important to know those specialized tissues. Now, I also want to remind you that you do need to be able to tell the difference between a root and a stem. And you can see here that the root diagram is very similar to a stem, because remember, a stem has those vascular bundles of tissues going around in a circle, and then they have got the cambium, which runs through it. It's like an inner circle. You can see the pictures look very, very similar. But the main difference is, remember those vascular bundles? They are divided into half. The top half having the phloem and the bottom half having the xylem. If we have a look at the diagram alongside, that is actually not the case. But before I get to those, let's make sure we start from the outside and work our way in. Now, I'm going to start off with these root hairs. Now, root hairs are really important because they increase the surface area. That is why we have them. And we need to increase the surface area so we can absorb more. Now, potentially, you will be asked to draw a root hair, and this is sort of what a root hair looks like. We've got our nucleus up in the top corner, and what makes root hairs unique is that they have a vacuole, which is what I'm drawing now, which goes the full length of your root hair. And the reason why you have the vacuole there is to create a osmotic difference. In other words, a difference in water, so that you're constantly pulling water in all the time. And remember, that's the main function of roots, is to anchor, but also to absorb water and minerals. Now, sitting just inside of that is, again, the cortex layer. And the cortex layer, remember, is made up of all of those ground tissues. Now, potentially there will be two kinds of ground tissues here. We'll have parenchyma, but we'll probably also have collenchyma too. Now, a quick side note, will we ever find chlorenchyma in a root? No, we will not, because roots do not have chloroplasts, because they cannot photosynthesize, because they are underground. So just keep that in mind. And actually, something I want to go back to before we go any further, I forgot to mention this about our root hairs. Our root hairs, they do not have a cuticle. So on the outside, there is no cuticle. Now, this makes sense because what do cuticles do? Cuticles waterproof. Now, why would you want to make the cell that is responsible for absorbing water waterproof? The answer, you wouldn't want to do that. You want your root hairs to absorb as much water as possible. So root hairs have no cuticle on them. Now let's move into our deeper layers, and we're going to go into the endodermis. Now, the endodermis is, as it suggests, endo means inside and dermis means skin. It is a layer that is very specially functioning to funnel water. In other words, cause water to flow in a certain direction. Now, what's not in this diagram, which we do need to know, which is found just below the endodermis or part of the endodermis, is something called the Casparian strip. Now, the Casparian strip is a waterproof layer that makes sure water gets where it needs to go. Now, again, I'm going to elaborate this more when we do transport of water in plants, but essentially, water is going to go from the outside soil into the root hair, through the cortex, through the endodermis, around the Casparian strip, and then into the xylem. And it's this Casparian strip that is waterproof, but it doesn't make sense now why there's a waterproof layer on the inside, but don't worry, the upcoming video will explain that too. We then have, sitting below that, as we've mentioned before, we have our xylem and we have our phloem. Xylem, there's our phloem, and you'll notice they're again in different locations. The phloem is now sitting on the center, and the xylem is in red, sitting in the right in the middle of our diagram and along the edges. Now, the last and final thing that I want to bring your attention to is something called the pericycle and also the vascular cambium. So I'm actually going to put these two together. The pericycle is again where we have growth and where we are going to affect water movement. And the vascular cambium, as we mentioned earlier, cambium means it is a meristem, it is growing, and it potentially will change the type of tissue it started off as. And so vascular cambium is essentially a meristem that turns into more vascular tissue. Now, as always, I like to finish off my lessons with terminology recap. Remember, you can use these to create flashcards, which makes studying so much easier, and you will definitely get more marks by using the correct terminology in your explanations. Now, as you can see, there was a lot of terminology, so let's quickly go through them all. We spoke about the tissue types in the beginning, and we spoke about the outer layer being called the epidermal tissue. This is the single layer of cells that sits on the outside of a plant. It's there for protection. protection, and it is specialized in leaves and in roots. We then spoke about ground tissue, which was that special packaging tissue that fills up the majority of the plant. It gives it shape, but also support. Examples of it we'll speak about just now, but they were things like sclerenchyma and collenchyma. We then spoke about vascular tissue, which was the xylem and phloem. They are the ones who are transporting substances within plants. We then moved on to specific structures like a cuticle. And a cuticle is a thin, waxy, waterproof layer that we find on the outside of leaves, sometimes stems, but never roots. Speaking of leaves, we moved on to the mesophyll layer, which is the middle layer of the leaf. It is where we find the specialized mesophyll palisade cells and the mesophyll spongy cells. Speaking of that packaging tissue, we looked at also mentioning the ground tissues, parenchyma, collenchyma, and sclerenchyma. Again, if you'd like more detail on that, go and watch my plant tissues video. We then moved on to regions or areas that you find in stems and roots. And that one region we spoke about a lot was the cortex. The cortex is this packaging layer. It is often right underneath the epidermal layer when we see this in stems and roots. Please don't think cortex, core. That's not what it means. We did just now mention xylem and phloem, so I'm not going to go over those again. Cambium, remember, cambium is a word to describe that very unique kind of tissue that can grow into almost anything. Cambium does fall under a non-permanent or semi-permanent tissue. It often allows for lateral growth and plants getting wider. Along with that, we also spoke about the pericycle, and the pericycle is closely related to the cambium. The pericycle is not a tissue, but rather a region. It's very similar to the cortex. It is an area or a layer, a name we give a layer. Speaking of layers, we also have the endodermis, which is the inner skin-like layer that we find in roots. It has a very important job of funneling water from the outside of the root to the core of the root, and it does this through the Casparian strip, which is this waterproof waxy layer right in the middle of the root, and it funnels water into xylem, which we'll get to in more detail at a later video. We also looked at root hairs and stomata, which were specialized epidermal cells that allowed substances into and out of a plant. Now, if you like this video, make sure you give it a thumbs up, and I will see you all again soon. Bye.